You're good to go.
Okay, and if I record that on this, though, will it record the audio if this is muted?
Is it the audio is coming out of here?
Okay, all right, so it's coming through.
Yeah.
All right. But it's okay.
Yeah.
All right, well, let's see how it goes. Yeah.
Oh.
Isn't that, it's not allowed me to minimize.
You won't be, you just have to put it to the side.
Okay. Right.
Actually, can I
Sure.
drag that back out?
Yeah.
Just hit that button. That's the button.
Yeah, I tried to do that.
It won't let you, automatically.
Maybe I'll just show the active speaker. Okay, that's good enough. Yeah,
Well, like you said, you can just drag it.
yeah, I can just drag it. Yeah. All right, thanks. Okay. Good. Got this thing here. I got this. Everyone seems like they're ready. All right. So this is the summer school. I have a little bit, I'm fighting a cold that I got a couple of days ago, but just came back from a meeting, the Organization for Human Brain Mapping in Bordeaux. So we were allowed to travel there and it worked out well, but I got a cold from it, or at least in the flight or something. So did everyone hear me well? Is it okay? So this course is kind of abbreviated from previous courses. It's the whole summer. It's twice a week. And you're welcome to come to any lecture you want to. It's not like a formal course or anything like that. And if you got my email, you'll see also that there is a Google Doc associated with notes with each lecture. So I just I populated with whatever I added to it, during my research for the lecture. I just put in papers and things like that. So you should have that there as well. So this is sort of a quick introduction to the course, but also I'd like to spend a little bit more time on just getting a perspective of the history of neuroimaging. So just to begin, okay. So this lecture, as I mentioned, is 18 lectures long, or this course is 18 lectures long, and it will cover everything from the basics of fMRI, MRI, some more advanced topics in fMRI like multi-echo fMRI, real-time fMRI with neurofeedback, resting state fMRI, which is a lot of fMRI connectivity mapping, different ways of processing the data, different ways of making sure the data are high quality, use of peripheral measures with fMRI, and then combining psychophysical or physiologic measures, and also different sort of more advanced topics still, looking at deep sampling, like averaging like crazy with fMRI, simultaneous EEG, physiology in the time series signal, and then high field strength and layer and column fMRI. And then finally, I'll end with a lecture on my perspective on the challenges and mysteries and controversies of fMRI that still exist, which is sort of a fun lecture that I like to give. I'll talk a little bit about some controversies today, just because I think it's kind of interesting. So I assume that a lot of you are either summer students or postbacks or some postdocs or whatever. So you may not have a big background in neuroimaging and fMRI, but I'm just going to start with just neuroimaging and go way back. So the first was, so the first type of information that we got from the brain, and like what parts of the brain did what, was starting to become more of a thing for neurologists in the late 1800s. And probably the most famous case, and one of the first cases, was this person named Phineas Gage. And he was a railroad worker, and he suffered the unfortunate accident of, while he was working on something, some dynamite went off and a beam that he's holding right there went through his eye and went through his frontal lobes. And miraculously, he didn't die. But what's interesting is that he seemed generally okay, except his whole personality changed. So, and this is according to the people that saw him, he became fitful, irreverent, impulsive, and prone to the grossest profanity with little respect for social norms, in contrast to his earlier reputation as a responsible, reliable foreman. So that was the first hint. It's not brain imaging, but if you damage a part of the brain, it's the first hint that frontal lobe is related to aspects of your personality and your judgment. So that was one indication. And there's been two other famous cases, situations where Paul Broca had a patient that could only produce the sounds tan, but he understood words perfectly. And it turns out when he died, he did a dissection, or he looked at his brain, and he found out this part of his brain in the anterior prefrontal areas, left prefrontal, is involved with word production. He assumed from that. And then another neurologist, Wernicke, so this was called Broca's area. Another neurologist, Wernicke, had a patient who had a lesion, who also, he could not understand or produce meaningful speech, but could articulate words perfectly well. But they were just nonsense. But he could at least articulate the words. So this area of the brain that was damaged was called Wernicke's area. That was associated then with understanding words or producing meaningful speech. So then, some information about the brain and approximate locations of what's important was being built. It's interesting. I just wanted to add this one paper that I just found this morning, actually. There was a nice review article on looking at disconnectivity. It's sort of an advanced field of considering disorders as a function of disconnectivity of white matter tracts. And they did an analysis of exactly where in the brain the white matter tracts were damaged for Leborn was the person who was only could make the words tan. And actually Henry Molson was another famous patient who had both of his hippocampi taken out. He couldn't remember anything after that. So it just basically shows, what parts of the brain were actually damaged with more detail. And it turns out not necessarily even the cortical areas are important, but also the connections. So the white matter connections are just as important. So that's a nice paper to take a look at. All right, the first official fMRI, or not fMRI, first official experiment that still wasn't quite neuroimaging, but it sort of was leading in that direction, was this work by Angelo Masso in the 1880s. He had this very delicate balance, and he lied a subject on the balance, and he had them do either nothing versus intense mental operations, and the balance supposedly would tip towards the head, and he inferred from that the brain sort of filled up with more blood as it was working harder. So that was one kind of crude but sort of clever inference back then. A paper that was just completely brilliant and was really prescient for all of fMRI, that is an amazing paper, was by Roy and Sherrington. And this was published way back in 1890. And there they had an animal, and they opened up their skull, and they were measuring something that was very sensitive to breathing, and also a measure related to blood pressure. And they gave a stimulation, like a weak electric stimulation to this central sciatic nerve. And in the area that they were looking, they found what looked amazingly like a hemodynamic response, like the types of hemodynamic responses we see today. So this was an amazing finding that kind of went ignored for 100 years. But it was literally 100 years ahead of its time in terms of looking at these very precisely timed blood pressure changes. And there was also a lot of quackery at the time, around 1900. Phrenology was a big thing. So that was the idea where people were thinking, okay, well, humans have big brains, and maybe if the part of the brain is bigger, that means you have more skills in certain areas. And so they divided the brain into all these segments that associated with personality or skills or other sort of things. And then they measured in detail your skull to determine the type of person you were. Obviously, it was nonsense, but they put a lot of work into it. But that was also going on. All right. So the back to the hard science. In 1895, William Röntgen discovered X-rays, and kind of by accident, and it's sort of a lesson in terms of how science is kind of actually how breakthroughs are actually in a lot of times how they happen. And he basically was looking at he had what's called a Crookes tube, which is a tube that's a vacuum, and he had a cathode and anode, and basically creating a potential between the two. And essentially, electrons would be accelerated towards the anode. And he was putting various materials on it, and he found that certain materials glowed. But then he found out that if he put something in front of those, he could actually see, the electrons penetrated softer materials, less dense materials, and didn't penetrate more dense materials. And he found that this was so on the glowing surface, it showed up as his skeleton. And that was a breakthrough, and we still use X-rays today. All right, so with X-rays, people wanted to do a little bit more. So they've went to extremes to look at to enhance the contrast with X-rays. They wanted to look at the brain. So they thought, okay, the CSF is getting in the way of looking at actually the brain to get at the brain contrast. So people back then sometimes had all the CSF drained from their brain so that they could just make a good image of the brain surface. That was pretty extreme. But the first angiogram also was created using X-ray. And this person, Antonio Egas Moniz, 1927, he was trying to come up with the right contrast agent that was dense enough to go in the vessels that blocked X-rays. And he first used strontium and lithium bromide, and he ended up killing a person trying that. So that didn't work. So he finally switched to 25% sodium iodide. And even today, we use iodine-based radiopaque contrast agents. So luckily, he switched that out. They didn't quite have, I guess, IRBs back then. You could just do whatever, apparently. So 1924, Hans Berger created the first electroencephalogram. He first measured alpha and beta waves. This was the setup. He basically had a few electrodes on the scalp, and he was able to detect these ongoing waves. So that was kind of useful, very noisy, of course, but useful. And then, okay, so then now we're going along, jump ahead to the 1950s. Some of the conceptual foundations for PET and SPECT were created in terms of trying to work out the math for doing tomographic reconstruction of, instead of just going through a certain, two-dimensional plane, the math was being worked out for having projection reconstruction, or with PET, sort of coincidence detection, and then projection reconstruction. So that was being done. And then, so finally in 1960, William Oldendorf patented an electronically based device that could capture slices continuously through a solid object. So this is called computed tomography, CT. And this is still used today for a lot of purposes. Hounsfield actually in 1971 finally implemented the first CT scanner. So that was in the early 70s, late 60s, early 70s. And now we're on to the function. So I remember actually when I was in grade school, I saw the Scientific American article, and it was actually very exciting for me. I mean, I was about 6th grade or 7th grade, but I remember seeing this and I got excited because this was one of the first functional brain images of a human taken sort of non-invasively. It was using CT, but with inhaled xenon-133. So actually, it wasn't CT. It was a scintillation counter that was basically, it wasn't CT. This was with a scintillation counter that was placed up to the side of the head that would basically count the radioactivity counts as it deposited in the brain. And it basically, they did the first types of experiments of reading, listening to words. What were the other ones? This is speaking. This is listening to words. This is, or actually speaking and then reading silently. So, and those brain images look pretty good. They're actually, it was kind of exciting that they could create this, but this quickly was surpassed by other techniques. So MEG came along. So David Cohen, 1950s, created the first MEG, magnetoencephalography device. Basically, initially, and so the concept of MEG is that you detect very, very weak magnetic fields on the surface of the skull that are produced by neurons that are sort of aligned in a certain direction. So axonal projections that create magnetic fields that wrap around on the surface of the skull. And so to detect those, initially copper induction coil was used that wasn't very sensitive. So James Zimmerman developed squid magnetometers, basically devices that were superconducting that were very sensitive to magnetic fields, very small magnetic field changes. So, and that was in the 70s. And this basically you needed a shielded room to do this. So this is a shield, and he put people in there. Now, of course, the EEG is a lot more advanced. You can have, hundreds and hundreds of electrodes on in your head. MEG is basically still using squids in this case with a hydrogen-cooled sort of dewar. Mostly, this is the dewar to cool the coils that detect the magnetic fields. Turns out that in 2006, and finally in 2010, it was demonstrated. We have optically pumped magnetometers, OPM, MEG, and we have an OPM right here at the NIH right now. We have both a squid and an OPM, and the idea there is kind of interesting. It's actually OPMs basically it's a laser that optically pumps alkali vapor, and it's such that the spins are aligned and they're polarized. And so if they experience a magnetic field, their polarization shifts a little bit. So their polarization, so it's pumped to be aligned, and then its polarization shifts when it detects a magnetic field. And as with MR, as we'll get into maybe in future lectures, the polarized spins persist at the Larmor frequency. And if these fields change slightly, then the precession frequency changes and the polarization shifts. And that's how it's detected by a subtle shift in the polarization of the light that passes through. So, and they're really nice because you can put them right up against the head, and they're very useful. So this is the concept I just explained. This is a standard squid device. The devices are a little bit further from the head, but that makes a huge difference in sensitivity. And here they're right up against the skull, and the sensitivity is much higher. And you can theoretically, walk around in this device. So it's sort of a big jump in MEG, and they're cheaper as well. All right. So now we're going to MRI. In the 50s, there was MRI was sort of something that was squarely in the domain of materials science and even used in oceanography to measure currents. But in samples, actually, Edwin Hahn discovered and explained a spin echo. So that was essentially where you give an RF pulse, and if you give an RF pulse to spins in a magnetic field that are processing at a certain frequency, they'll become just naturally dephased because the field homogeneities sort of cause them to dephase, and so the signal runs out. And this is what physicists were up against, sort of this signal decay, pretty rapid. And then what would happen is that, so what Hahn discovered is that if you gave a 180-degree pulse, you basically switched the precession of the spins, made them exactly opposite of what they were. And so they came back into focus. And that was called the Hahn spin echo. And that was very useful for measuring, increasing the timing, the time at which you can make measurements with MRI. But it was also, you could do a lot of things with that extra time to sort of make use of that signal. And what you did with the extra time, one of the things you could do is make images. So in 1973, so it took about 50 years or 20 years from the 50s to the early 70s for Paul Lauterbur and Peter Mansfield. So they were doing very different things. Paul Lauterbur was developed a technique for spatial encoding called zoomatography. And that was basically, if you give an RF pulse to spins that are precessing in a magnet, and you apply gradients, the gradients actually have there's a magnetic field now that's in proportion to where it's located. So there's a weaker magnetic field on one side of the board, a higher magnetic field on the other. So you can spatially encode the spins by causing them to precess at different frequencies, depending on where they are in the board. So he also, there's ways, and we'll get into that, I think, a little bit with talking about the basics of MRI. But he actually invented slice selection. And so actually, no, Peter Mansfield. No, actually, he invented slice selection with zoomatography. Peter Mansfield did a lot of the formalism. He worked out the math of K-space, so-called. People always refer to K-space as, where the way you process through this gradient space to encode the information to create an image. He worked out the math for that. He worked out phase and frequency encoding. Actually, he also did work out slice selection. So Lauterbur had a method for doing imaging, but it never really caught on, but it was using gradients. The concept was there, whereas Peter Mansfield's method is what we use today. And Peter Mansfield was pretty much ahead of his time. He came up with the idea of what's called echoplanar imaging that we use for fMRI today. And he came up with it even though none of the gradients were strong enough to actually implement, to be able to switch fast enough to do what he wanted them to do. So we have slower imaging techniques for standard imaging and echoplanar for very, very high speed. And so basically the idea is that you do all your spatial encoding by switching your gradients very rapidly with one echo, with one either spin echo or gradient echo. And so that was Peter Mansfield's contribution. So, and they won the Nobel Prize in 2003. So there's one person, this is actually the idea. So Paul Lauterbur had zoomatography. It was basically a projection reconstruction technique with gradients that never quite caught on. And then Peter Mansfield technique of slice selection that caught on much better. I won't get into those slides, but there was another person who didn't win the Nobel Prize, Raymond Damadian. And he actually did a lot of good work, but he was a little bit outside the field, and he actually he had the same idea. He created an MR scanner, and it was mostly, he didn't really hit upon the idea of using gradients, magnetic field gradients, to encode the location. He actually had what was called point process MRI, where he basically collected a data point, moved the subject, collect another data point, move the subject, collect another data point, and so on. So, and he laboriously made his images that way. So it's much less efficient. And, he was sort of an abrasive person. So, maybe because he didn't win, who knows, maybe he never felt like he got enough credit. But maybe he did deserve the Nobel Prize along with them, but we'll never know. This is the sensitive point method that he created around with. So okay, now comes along positron emission tomography. So that was invented in 1975 and it had commercial use in the 1980s. And so you should notice also, I'm not really going into the details of the technique. I'm just giving like an overview of the methods as they came along for the most part. Positron emission tomography basically does use radio tracers and radioactive tracers that are either, bound to some ligand that's either deposits itself in the brain like dopamine or whatever. And it relies on coincidence detection. And it detects positrons that are emitted from these radioactive tracers. And it relies on the fact that they're detected at the same time. And then it does projection reconstruction to recreate where that source came from. And it measures the density of wherever these radioactive traces are emitted. So it's a very nice technique for localizing in 3 dimensions radioactive tracers. So this was a technique that was inspiring as well. Came out in 1988, I think was the first paper in Nature with Steve Peterson from WashU and the whole group there, Mark Rakel, Peter Fox. And, they had these first iconic images of hearing words, seeing words. I think they wanted to copy what was produced in Scientific American by Martin Ingvar with Xenon CT. And their images are better and they're in 3 dimensions. But this takes a long time for the images for the tracer to accumulate. And of course, it's invasive because it's radioactive and the machines are very expensive. So this actually is a nice timeline. I'm not going to go over the entire timeline, but so essentially 1975, the PET scanner was invented. A lot of really good work on looking at either metabolism or blood flow with various tracers was developed from the 70s to the 80s. Rakel had a famous paper looking at oxygen consumption and blood volume in 1988. And finally, there's now PET CT hybrids and there's PET MR hybrids as well for looking at the human brain. So, right. So another technique that sort of came along about the same time as fMRI is near-infrared spectroscopy. And even though conceptually it was sort of developed in the late 70s, finally about 1985, there were clinical studies on newborns. 1989, the first commercial use of near-infrared. And so the basic idea, and then also 1991 and 92, the first studies were carried out by Britton Chance, Arno Willinger, and a few other people using single-channel instruments. And near-infrared actually measures the reflectance of light. So you have these probes that could be on the brain or anywhere in the body that has a source of infrared light, and it looks at the reflectance. It measures that with the receiver. So it doesn't penetrate too deeply. And the idea is that blood has a certain reflectance. And if it's oxygenated, its reflectance changes. So you can measure the amount of blood or the reflectance of the blood, depending, looking at the spectrum here. And just to go into a little bit more detail here on the mechanism, because it does relate to fMRI a lot. So here's basically looking at the wavelength of light that's reflected, and this is the difference in oxygenation. So, and this is the absorption, this is the reflectance, or actually the absorption of the light. So you can see when blood is 100% oxygen, it's not very absorbed, and then finally, when it's 0% oxygen, it highly absorbs light. So on this end of the spectrum, blood is extremely sensitive, or the absorption is extremely sensitive to blood oxygenation. And then right here, which is called the isobestic point, which is about 800 nanometers, there is no change. So it's only, this change means there's a change in blood volume. So when that point changes, and then it flips. Actually, it turns out that the relationship between absorption and reflectance changes. It flips to the opposite side above 800 nanometers. So there's been some basic studies of looking at, with either breathing 20% air versus 10%, you see a shift. And this is actually looking at a evoked response with brain activation. And what people actually found, this is kind of a nice confirmation of bold effects, which I'll get into in a second, but basically saying that measuring oxygenation, total hemoglobin, and deoxyhemoglobin, and looking at the time courses. And you can see that there's a difference. And it turns out the deoxyhemoglobin is delayed from the oxy and the total hemoglobin. That's basically, there's this inflow of blood that's oxygenated, and then downstream, the deoxygenated blood goes down. So this is flipped. Actually, the deoxygenated blood actually goes down, but the signal is rectified to compare the timings. And that matches the bold signal quite nicely. And this is from this paper right here from Hubert in 2005. So, there's many commercial systems available now. It's kind of nice because you can actually just stick it on your head and wear it around. And okay, so that leads us to MRI. So, so MRI came along as I mentioned, that it was developed. The pioneers were Lauterbur and Mansfield, and it was invented in '77. That's the time that I think the patent was put in for it. It was clinically introduced about 1984, about the same time as the Apple computer, and it quickly caught on. There were scanners all over the world almost immediately because it was so useful for looking at soft tissue and lesions and tumors and stroke in the brain and hemorrhages. And so essentially the magnet contains many different things. There's the basically superconducting wire to make the magnetic field. To keep the wire cool, you have a dewar of liquid helium. And then inside is the gradient coils that spatially encode the images. And then you have a radio frequency coil. So when the spins are inside the magnet, at either 1.5 or 3 Tesla, they're processing, or the water spins are processing at a frequency that's approximately, well, in this case, at 3 Tesla, it's 127 megahertz, which is like a radio frequency. And so that's why you need radio frequency coils. So these coils give energy to the system, and then they receive these echoes, these Hahn spin echoes or other types of echoes, back from the system while the spins are being encoded in space. And so that's what the radio frequency coil is for. It gives the energy to the system and receives it back. And this is another view of it, more of a schematized view of everything that's involved. You need also amplifiers for the gradient. You need obviously a computer to control the gradients. You need a digitizer for receiving the signal, something for executing the pulse program, the pulse sequence, and so on. So this is a very basic layout. All right, so these are all the types of images that can be produced with MRI right now. So there's and it's also all of these images, and I'm not going to go into any detail as to how these images are created because this would be an entire course in itself. But venography, you can be sensitized to flowing blood, the blood flow. You can be sensitized to tissue characteristics. It's what's called T1 or T2 weighted images that are longitudinal relaxation. You can be sensitized to fiber tracks. And you can be sensitive to perfusion, which is, tiny vessels perfusing into areas. And you can slice and dice this any way you want. You can 3D project it. This actually happens to be my brain, just projected in 3 dimensions, just basically rotating around. And you can do whatever. And so now we're getting to the first fMRI experiment. So the first fMRI experiment was actually not bold contact. It wasn't using oxygenation. That wasn't using fMRI as we know it today. It was actually kind of a one-off study by Jack Beliveau at Mass General Hospital. And it was published in Science in 1991. And the idea there was that he used echoplanar imaging, this high-speed technique that collected one image pretty much every second. And he injected a contrast agent. This wasn't like the type that the X-ray doesn't see. This is basically a contrast agent that causes magnetic field changes that distort the signal, that causes signal drop as it passes through each voxel. And so to the degree it passes through is a measure of the blood volume in each voxel. So he first made one injection when the subject was not looking at a stimulus, and then he made another injection when the subject was looking at the stimulus, just a flashing checkerboard, subtracted these two images that were created by integrating over this curve and made a nice map of blood volume changes with two sequential ejections of gadolinium. So this was sort of a breakthrough. Everyone was incredibly excited, but as I like to say, even before the ink on the Science article dried, his colleague Ken Kwong came along and demonstrated that you don't even need contrast agents. So it turns out that blood is very much like gadolinium, and in the sense that because when it's deoxygenated, it has free electrons, and that actually causes magnetic field distortions around red blood cells or red blood vessels. But when it's fully oxygenated, it's diamagnetic like the rest of tissue, so that the fields smooth out. So you have a lower field when it's deoxygenated. When the blood becomes more oxygenated, the field becomes more smoothed out, the signal goes out. And anyway, this was Jack Beliveau's iconic science paper. So with blood oxidation-dependent contrast, you can trace back the origins back to Linus Pauling in 1936, who first studied blood susceptibility changes with oxygenation. So that was known since 1936 with Linus Pauling. And then three pretty important papers. Keith Philborn measured the T2, like the transverse relaxation properties of blood. This was in 1982. That was 10 years before fMRI began. Seiji Ogawa was the first person to show blood oxidation contrasts in animals. And also he did it in other test tubes and whatever. And then Bob Turner, when he was here at the NIH, he actually modulated, he cut off oxygen. He caused cat apnea, essentially. Cut off oxygen to cat brains, showed the signal go down as the blood oxidation went down, gave them oxygen again, and then the signal went back up. So, and so the basic idea is that, once again, if you can actually picture magnetic field lines passing through an oxygenated red blood cell, and everything is clean. The red blood cells look like the rest of tissue, they're diamagnetic. But when they're deoxygenated, they concentrate the magnetic field, and that causes distortions around the red blood cells. And because the precession of individual water molecules is exactly proportional to the magnetic field that they're experiencing, the water molecules will quickly get out of phase. They'll precess at different frequencies, and within that voxel, they'll dephase and the signal will go down. So that's the basic idea of bold contrast. So with Linus Pauling, he even said he even traced it back to Michael Faraday in 1845. So he even said, well, if Michael Faraday measured the blood in suspension, he would have found that the susceptibility changed with oxygenation, but he didn't do that. So he started with Michael Faraday, and then he showed that susceptibility changed with oxygenation. And as I mentioned, Pete Philborn measured this T2 relationship, this very nice relationship between the T2 of blood and oxygenation. And it's interesting because when that happened, everyone saw that it was proportional to oxygenation, but they didn't think it was enough. They actually, going back to the field back then, they thought, well, it's something, but it's, we don't know. And this is the problem with fields of science that are kind of siloed in some sense, because we were all physicists working on this sort of thing. We're thinking, okay, how can you use it clinically? Maybe we can look at hemorrhaging or something like that. But we didn't even think that with brain activation, the oxygenation would actually change with activation. So that didn't occur to anyone for 10 years. So Seiji Ogawa came along, and he actually showed pretty nicely that in animal brains, breathing 100% oxygen versus breathing 20% oxygen, so he over-oxygenated their vessels versus having normal room air. And you can see where all the veins are that pop out when they're becoming deoxygenated as opposed to hyperoxygenated. And he showed that in test tubes as well. This is 0 oxygenated blood, 100% oxygenated blood. Blood is still dark, but there's not this penumbra that dephases the signal. And So actually Ogawa predicted fMRI, but being a physicist, he actually, and nobody really knew either. I mean, he predicted, he's like, well, we could expect this oxidation sensitive contrast could be used to monitor regional oxidation changes in the brain. When some region of the brain is much more active than others, the active region should show darker lines in the image because of the increased level of deoxyhemoglobin resulting from increased oxygen consumption. So he knew something would change, but he just didn't get the sign right. When in fact, the opposite happens, which is still paradoxical to some degree. So when you have brain activation, the flow goes up, the oxygenation actually increases, it overcompensates for the metabolic rate change. The reason for that is both to deliver oxygen, of course, but also to remove waste. And I think that the field is shifting in terms of where they're emphasizing that. I think that, even without a flow response, the brain would work just fine with no flow response because there's more than enough oxygen to be delivered. But if the metabolic waste products stay in the blood or in the tissue, then you have problems. So now people are beginning to feel that the blood flow response is mostly to remove waste as opposed to deliver oxygen. So anyway, but Ogawa had the right idea and just had the wrong sign, quickly corrected his problem when he talked to some neurophysiologists. But even the neurophysiologists didn't know this. And I'll get into this. So then he said, therefore, in addition to the anatomy of the brain, one aspect of physiology could be studied by the MRI of water. And so this is another depiction of the field distortions around a vessel, like basically what they look like. It's a dipole. It's a quadrupole right here. So anywhere where there's distortions, there's spins processing at different frequencies that get out of phase and cancel. So Bob Turner, as I mentioned, did his cat study and showed pretty dramatically that when you cut off the oxygen from the cat brain, the signal goes down rapidly. When you return it, it goes back up and it overshoots. So by now the field was really excited. And so this was the experiment that I remember seeing this movie when I was a graduate student at ISMRM in August 12, 1991. And yeah, my jaw just dropped. This is, Ken Kwong didn't show it. Actually, Tom Brady, who was in charge of the NMR Center, showed it during his plenary lecture. Didn't really fit into the rest of his lecture, but he just showed it because it was new data. And so he showed this brain activation change right here. And the entire audience was like, what the heck? What is that? This is amazing. So Ken's paper was, Ken was the first to do the experiment, produced a paper in 1982 showing this. He did a lot of other work. He did different measures of perfusion as well. And he compared it with PET studies, looking at different frequencies of checkerboards. And this is his notebook. He shared this with us. And that just shows the date of the first experiment for, if anyone cares, looks like May 9th, 1991. And he had a block design experiment and he was using bold contrast. The echo time was 40 and I'm sorry, 40 centimeters. And yeah. Anyway. And that's also the inversion recovery. That was CBF contrast. So another group was doing this as well. A group in Minnesota. And they actually were a little bit delayed because they thought they had to wait. And it always mystifies me when I talk to them. They thought they had to wait until they got a 4-Tesla scanner to actually get successful results, which is not true at all. You could have done this at 1.5, but they got their 4-Tesla scanner. They didn't use echoplanar imaging, but they did do something that was good and novel. They actually compared the echo times. They showed that it was bold contrast because the idea that with shorter echo times, there was less time for spins that are experiencing different magnetic fields to go out of phase. And so there would be less of an effect with brain activation. And with a longer echo time, there'd be more of an effect. So they showed this echo time effect, which was really nice. A nice demonstration that it's a bold contrast. And they showed visual stimulation, and it was a little bit higher resolution. So it was a nice study. But the workhorse experiment, though, was echoplanar imaging, single shot echoplanar imaging. They're basically, when you give an RF pulse, the signal decays, and it takes about 40 milliseconds, before it's or 50 milliseconds before it's gone. With echoplanar imaging, to encode even a low resolution image, it takes about 20 milliseconds or maybe up to 40 milliseconds at that time. And so you need to oscillate your gradients as rapidly as possible to do spatial encoding before the signal decays away. And so for fast imaging, MGH had this retrofitted system that was a resonant gradient system. Minnesota had a standard gradients with the multi-shot technique, which is easier but noisier. And I was lucky enough to be part of a group that used working with Eric Wong, who built as part of his thesis was local head grading coils that were very low inductance, so they could actually switch very quickly. And so that's what we used. So we were like after I saw Tom Brady's talk, we had already built this coil for other purposes. And basically this is Eric's setup. We had a coil in the scanner. It was basically sewer pipe, epoxy, and wire. And it was a really fun experience putting this together with Eric. Before the meeting, he wanted to do echoplanar to demonstrate his perfusion technique, which didn't work well. But we used it for this purpose. There's two versions right here. One thing also, another example of IRB, this would never be approved today. This coil, just putting it in the scanner, and because if it's shorted out, it would like torque, and that would be very dangerous. And so things that we did back then were, it was great that we were allowed to do it, but we were lucky, one, that no one got hurt, and two, that that everything worked out fine, and it was fine. Eric knew what he was doing. I didn't really know what I was doing. Eric actually knew what he was doing. And he basically designed this coil using a technique called conjugate gradient descent that optimized for the field. And this is what it looks like. We basically took over the machine shop and built this coil. And this is us just sort of building it in the middle of the night. We used the lathe to sort of build grooves. This is the Z-axis. And then on top of that, we put the X and Y axis. With more epoxy, we traced the patterns in, and then we used a Dremel tool to groove in the grooves of the wires, and then pounded the wires in with a hammer, and repeated this until we had X, Y, and Z coils. And we also had to build RF coils from scratch too. Basically several different types of RF coils. And our first experiment was we'd only collect one slice at a time. We had 2.5 centimeter width, which was extremely large, extremely thick. And we both realized after the fact that we didn't even need a gradient coil to do this. I mean, literally every scanner in the country, in the world, could have done echoplanar imaging, functional MRI. They just had to go to a little bit lower resolution, 5 by 5 by 5 millimeters, which is considered crazy big today, but it was still better than PET scanning back then. And that was quite easy to do with our wimpy 100 amp gradient amplifiers back then. So every place could have done it. They could have done echoplanar imaging, they could have done fMRI and produced great results. But we felt we had to build a gradient coil. We got a little bit higher resolution, and we did it. And these were our results. We actually, so we were the third group to actually produce results, and we got them published. So that was my notebook. Our first study was in September of 1991. So here's what it looks like. So this is one of our first movies of, one thing that was good about what our group did that helped prove that the whole time at this time, you had to prove that the effect was real. So the way to do this was either, as the Minnesota group, they showed the echo time dependence. As Ken Kwong did, he compared to PET with different stimulus frequencies. What we did motor cortex, we did left versus right motor cortex, looking at the whole brain. So that wasn't just like one side, it was actually showing both motor cortex, like left, right. And so this was the study there. And this is the results right here. Basically, the hemodynamic response takes about 8 to 10 seconds to plateau in the on state, and it takes a little bit longer to turn on. And so the first thing that we thought when we did this, because remember, the whole time I was, based on Ogawa's paper and based on not reading the literature that much about physiology, I thought, well, I was looking for a signal to go down. In fact, in my notebook, I had a question mark and an exclamation point because the signal kept on going up with activation. I was expecting it to go down, but it went up. And so I dug in the literature and I found two really good papers that actually explained why the signal should go up. That were there in the literature since 1986, at least for Peter Fox. Here was a PET study that showed flow, basically showed flow, metabolic rate, and compared the two to get a measure of oxygen extraction fraction. And he found that with activation, the extraction fraction went down by about 20%, which means that the blood oxygenation should go up. So that was a really, really valuable paper. This was fundamental for getting any of the papers out, to have at least some mechanism that would explain this. And there was actually another paper, a kind of a nice paper by Frostig that showed, and this is actually using a slightly different range of frequencies. This is the UV's visible spectrum. That actually showed, this was actually a demonstration of the hemodynamic response with visual stimulation. He was also showing oxygen columns, but here he just showed just the reflectance changes of the blood and looking at blood volume and blood volume and blood oxygenation. So that actually came out in 1990. So those were already out when we were trying to write up our papers. So we found these papers, referred to them, and were able to write it up. So the basic idea, as I said before, is that during rest, you have, the vessels are constricted or less dilated, and you have deoxygenated hemoglobin, a little bit of oxygenated hemoglobin as well. And then with activation, blood rushes in, it becomes more oxygenated. There's less distortions, longer T2, higher signal. And this is the whole mechanism right here. And I'm not going to go into too much depth. Basically, activation causes an increased arterial blood flow and volume, where the oxygen delivery exceeds the metabolic need. This increases the capillary and venous oxygenation. It decreases the amount of deoxyhemoglobin. So what we care about is the deoxyhemoglobin. That's the contrast agent. So if we have less of that, you have a higher signal. The oxyhemoglobin, if we have more or less of that, it doesn't make a difference because that's sort of invisible. That's looks like the rest of tissue. But the deoxyhemoglobin is the contrast agent. And if you increase or decrease that, that's what matters. So then you have a decrease in susceptibility related to dephasing, an increase in what's called T2 or T2 star, and then the signal goes up when you have sequenced it or weighted towards that. So there was a coupling controversy, actually, even after the results were produced and even after the papers were published. There was this big, it's kind of a fun paper to read by Peter Fox back in 2012 in Neuroimaging. There's a special issue on 20 years of fMRI. And Sokolov actually was a person here at the NIH. And I remember coming here when I was even first, applying for my job, or maybe when I first arrived or something. And he got up and challenged me on why I think that fMRI is meaningful or something like that. He basically said, how do you know that that's just not, vessel squeezing or some other thing? Why is it oxidation increase? And so the idea here is this, and he had a really well-considered point. It's that he did PET and metabolic rate changes, and he actually thought that the flow was in response to metabolic rate, and that it should be tightly coupled. The amount of flow should be exactly match the metabolic rate. So you shouldn't really see any oxidation change. It just should match it perfectly. And that makes sense. I mean, there's no reason to think that shouldn't be that case. And so what Fox argued with his PET paper that he produced in '86, and with further evidence from fMRI, that there was this decoupling, that the flow was overshooting the need for oxygen to increase the metabolic rate. Metabolic rate only goes up a little bit. So, and it's ironic because in some sense, Sokolov might end up being right in this regard. I mean, he's not right in that fMRI is meaningless. He's might be right in terms, as I mentioned before, of the reason for flow to go up. It's not just to match. If it were just to match the metabolic need, it might just not go up that much, and the oxygenation wouldn't go up that much. But if it's to remove waste, then you might need an overshoot to normalize the pH and to get rid of CO2. So anyway, so we might end up being right. So in 1992, there were 5 bold papers produced. What time is it? Let's see here. Okay, I'm just about done. 5 papers produced. As I mentioned, each had their own unique characteristics. And I only have like 3 slides left, or 3 or 4, I think. Activation statistics, you'll get into this more than enough later on in the courses. Basically, there's various ways of extracting significant activation based on features of the time course from the data. And so now fMRI is the big method that complements all these other methods with MRI. And as I mentioned, the first studies in fMRI it was like the Wild West. You know, we did visual, auditory, motor, we looked at the dynamics, we tried to model it. We did parametric variation of the intensity to compare it with psychophysical results. We looked at the pulse sequence sensitivity, resolution comparisons, and the noise, which led to resting state fMRI. And we tried to modulate the signal by CO2, O2, breath holds. And so there are all kinds of experiments that were in the early days. And they're still going on today. This is just some work from my thesis work. I was trying to modulate the signal, see how fast it can go and keep up. I tried to see how short of a stimulus you can give and still see a signal. Tried to increase the resolution, see how the signal changed. Modulated the oxygenation of the blood. Even scanned a person for like 25 minutes continuously to make sure the signal stayed up. So we did all kinds of things, which was a lot of fun. And there was parametric studies too. This is some work by the Medical College of Wisconsin. Steve Rayo was doing motor cortex tapping at different rates, which looks like a very, very straight line. It looks like he actually just made up this data, but it's not, I think, even though there's no error bars, but oh well, it's okay. That's how things were back then. It's like, but then this is also a different study with auditory cortex with syllable rate. It seemed to, in the auditory cortex, showed monotonic response characteristic. And Steve Engel kind of blew everyone's mind when he did a study where he had a visual stimulus that was increasing in eccentricity that basically was a ring that just kept on increasing. And so he was able to create bold ripples in the visual cortex. And these ripples were very much, it was basically what was active when the ring kept on expanding. And the size of the ripples gave a measure of the accuracy of the bold response. And then from there, the vision scientists took hold of this and started mapping out visual cortex, looking at from not only from doing eccentricity, but also doing a rotating wedge to get at where the borders of the visual system are, because the way that the signal propagates when you have a rotating wedge actually is mirror imaged across each visual area. So if the signal actually starts to go towards itself, and that border is a border of like visual V1 and V2. If it's going away, then that's V2 to V3. So you can actually measure the visual borders by the way the signal is propagating as you're rotating this wedge. So they were very clever. They made nice maps of V1, V2, and V3, and so on. And so, the brain is really complex. And the ongoing question now is, how useful is BOLD? How useful is fMRI to understand the brain? And, it has many functions. This is the temporal and spatial resolution. And so this diagram, I think, is one of my last ones, basically showing that fMRI can pretty much cover everything from 1 millimeter, everything from circuits to the whole brain, which circuits means like down to layers and columns, which gives you information about how the brain is organized. It may not give you, fundamental information about, how individual neurons communicate, but as far as we know, the smallest clusters of neurons are either layers or columns. So that might be good enough in terms of understanding mechanisms of how the brain processes information. In terms of time, it's extremely useful. Certainly most neural activity at the very low level occurs very rapidly, so we can't get at that. But most neural activity relevant to human behavior, most human behavior is, over the time course of seconds to days, which fMRI covers just fine. We process information over seconds. Our cognitive process, our memory, neurotransmitters, hormones, learning and plasticity, is all timescales that fMRI can capture perfectly. So, and there's several pillars. I'll talk about this next lecture, basic aspects of fMRI. I'll get into more detail. And given the shortness of time, I'm not going to go into it. And actually, this is the last one. Basically, a time course of, or a timeline of all the major achievements or things that went on in fMRI. And there were a lot of controversies, as you can see. There's about 10 or 15 of these. And a lot of advancements. The field was busy all the way up till now. So I might get into that a little bit, but I'm not going to talk about it obviously now. So with that, I'd just like to end it. So, and also these slides can be, if you want them, just ask me. I'll make them available, I don't know, on the Google Slides or something like that. So, all right, thanks. So sorry, that was a little bit long, but any questions? All right, well, that's okay. Take a look at the notes. If you want my slides, like I said, I'll put a link to the slides in the notes. So, all right.
Hi.
Hello.
My last week.
Oh, hi.
My something else.
Oh, yeah. Yes. Yeah, you're here.
Yeah,
You're here.
exactly.
So, who are you?
Tanya.
Oh, okay. That's right. Yeah, I remember seeing your email.
Yeah,
That's
yeah.
right. I'm glad you're here. Awesome.
Finally.
Yeah, great. Yeah, for some reason I thought I recognized you. I don't know why, but yeah. It's,
Yeah.
different context.
I saw that you had a delay in my face and
Yeah.
maybe.
Yeah, I was trying to think. But anyway, yeah.
So I had a question about you said
Thank you.
the lot and the oxygenation. The oxygenation is not only about that the brain needs oxygen.
Right.
It's about, yeah.
That's kind of controversial. I mean, I might be a little bit in the minority still in terms of, it turns out there's studies done where they stopped. They actually stopped the flow response and they gave neural activation. And the oxidation went down in the blood a little bit, but the neurons were totally fine. The blood flow was still going on, but it was sort of like a baseline blood flow. It didn't go up. And the neurons were happily firing. There was no compromise in neural activity. So that seems to suggest that it's not that important that flow goes up to supply oxygen. The thought is though that possibly, long-term stimulation, metabolic rate, waste might accumulate, and so there might be like a longer sort of degradation. But there's been recent papers that suggest that flow responses mostly to keep CO2 levels and pH levels constant. If those accumulate, it's like just damages tissue. But the lack of oxygen, and also there's another clue to this, is that if you give low oxygen, you don't get a flow response. You're like breathing like 5% oxygen. You might feel like you can't breathe that much, but you won't see, there won't be any flow response to compensate.
I think they have.
Oh, okay, yeah. Right. But if you breathe CO2, you get a large flow response, which is CO2 is a metabolic product of the brain. So that's sort of a hint of that.
Thanks for the lecture.
Oh, thank you. Yeah, I still have to take a look at your paper.
It's a really long and dense, and I'm sorry.
Yeah, well, that's okay. I'm trying to get my head around. But,
I'm actually trying to make a new paper that's more application focused.